In this video, we are going to discuss how the structure of the eye allows us to process images. Remember, the eye is about the size of a ping pong ball. It has a pupil, which has pupillary muscles that control the amount of light that enters the eye. This light goes through the pupil and hits the lens, which is held in place by the ciliary bodies. The ciliary bodies have muscles that either contract or relax to change the shape of the lens in order to allow it to focus on the retina. The inner layer of the eye is this choroid, which has blood vessels that nourish the eye. And then our last layer of the eye is this retina, which contains the pigments and nerve cells that allow us to transform that light into electrical impulses that can be understood by the brain. So let's take a closer look at the retina. This right here is a magnified view of the retina. This first layer right here is a pigmented part. The excess light not absorbed by the rods and cones is absorbed by the pigments in the pigmented layer. Here are the rods and cones, which also contain pigments. The cones have a pigment called cone opsin, also known as iodopsin, and the rods have a similar but different pigment called rhodopsin. And these pigments are important because when they break down, they allow for the eventual creation of neural impulses. These are electrical impulses. And what happens first is that the rods and cones synapse onto the bipolar cells, here in purple. And then the bipolar cells in turn synapse onto the ganglia. Ganglia is plural for ganglion, and these are also nerve cells. And remember that in addition to the ganglia, we have these amacrine cells, which help regulate the sensitivity. They regulate these neural impulses. And so the most important takeaway from this is that there are two synapses that occur. Our first synapse is here between the cones and the bipolar cells, or the rods and the bipolar cells. And the second synapse occurs between the bipolar cells and the ganglia. And finally, the ganglia synapse onto the optic disc, which leads to the optic nerve. Now, of course, we have two eyes. Each eye has an optic disc, and each eye has their own optic nerve. These two optic nerves have a crossover point called the optic chiasm. This crossover point is very important for this reason. I want you to do this exercise with me. Think about your two eyes. You have your left eye, your right eye. Now what I need you to do is I need you to close your right eye and look with your left eye. What is the furthest object you can see to the left with your left eye? What is the furthest object you can see to your right? Now close your left eye, open your right eye, and repeat the procedure. What is the furthest left object you can see with your right eye? And what is the furthest right object that you can see with your right eye? Open your eyes and look at this diagram. Right here, as you can see that I've demonstrated, in your left field of vision, this red area, there is an overlap with what your right eye is able to see. Similarly, your left eye is able to see part of your right field of vision. So the information that your left eye sends down your optic nerve is your left field of vision as well as your right field of vision. Similarly, your right eye contains information from your right field of vision and your left field of vision. So what happens at the optic chiasm is that this information is shuttled to the proper nerve. The information for your left field travels down the left optic nerve, and the information for your right field travels down this right optic nerve. Ultimately, these optic nerve synapse upon the lateral geniculate nuclei. Geniculate comes from the word knee in Latin, so don't really worry about what geniculate means, because the scientists that named this thought that the nuclei looked like knees. Remember, nuclei is plural for nucleus. From here, the lateral geniculate nuclei synapse upon the visual cortex. What is tricky is that the left optic nerve synapses onto the right lateral geniculate nucleus, and the right optic nerve synapses onto the left lateral geniculate nucleus. The right lateral geniculate nucleus then synapses onto the right part of the visual cortex, and the left lateral geniculate nucleus then synapses onto the left area of the visual cortex. When it comes to vision, we are capable of parallel processing. Parallel processing essentially means that we are able to multitask when it comes to our vision. It's not like baking, where you start off putting flour and then adding eggs to the bowl and continuing with the recipe one step at a time. Parallel processing, in the sense of our vision, means that we can see things such as color, movement, depth perception, that is, how close something is to you, how far another object is away. All of these things we can see practically immediately. This is what is known as parallel processing. Let's look at how we perceive color vision. As I said earlier, we have cones in our retina. These cones are capable of color vision. The cones have a pigment called cone opsin, also known as iodopsin. There are three types of cones. There is a red cone, a green cone, and a blue cone. And there are many red, green, and blue cones in the retina. Now, if you recall back to your primary school days, I'm sure you remember learning that red and blue can be mixed together to make purple. Simple enough, right? However, with color vision, that is not the case. So, if you want to perceive yellow, what is going to happen is that your red cones, your green cones, and your blue cones are going to be stimulated to different extents. So, to see yellow, your green cones are going to be highly stimulated, your red cones are going to be moderately stimulated, and your blue cones are either barely going to be stimulated or not at all. So, essentially, different combinations of these three cones allow us to perceive different colors, and it's not simply as intuitive as mixing watercolors. Let's review the visual pathway. What I'm going to do is I'm going to draw a small little eye in the corner here so that you can follow along while we go through these steps. Remember, you have your eye with the pupil, which controls the amount of light that enters the eye. Here, you have your ciliary bodies, which contain those ciliary muscles that either contract or relax to change the shape of the lens. You have the choroid, which has your blood vessels in the eye that nourish it. And finally, you have this retina, which contains the pigments and nerve cells responsible to allow you to transfer information to your brain via that optic disc and ultimately the optic nerve. First, light enters the eye at the pupil. Then this light hits the lens, which refracts the light back towards the retina. Within the retina, first the light hits that pigmented portion of the retina. Remember, those pigments allow us to absorb photons that the rods and cones are not going to absorb. So the rods and cones are also going to absorb light, and this absorption of light allows the creation of an electrical impulse, which is sent to the bipolar cells, those nerve cells. From here, the bipolar cells synapse onto the ganglia and the amacrine cells. And finally, the ganglia synapse onto the optic disc. Now, this neural impulse leaves the eye, travels down the axon of the optic nerves, because remember, we have one nerve for each eye. This impulse reaches the optic chiasm, that's that crossover. The information for each field is transferred at this point and carries down its respective optic nerve to the lateral geniculate nuclei. The left eye goes to the right lateral geniculate nuclei. The right eye optic nerve goes to the left lateral geniculate nuclei. And then finally, all of this information ends up in the visual cortex.